So here I'd like to very brief show you how to detecting recombination, which is also quite common if you are studying virus, for example. So usually you have several different methods to detect if a particular sequence have recombinations, and you can base on the similarity, just based on if they are similar to each other, or based on the distance method is called a recombination analysis tool, and I will see that in a minute. Or you can just compare different phylogeny or use the compatibility test to see if they have different recombinations. And this case is from the BBTV. It's a banana bunch top virus, and this is from my own study. And this virus is kind of very strange. It has a unique. It has a 6 component, and the 6 component share a stem loop region and also the 3' comma region, but they differ on the other coding region. So there's something interesting going on, that all the different components share something, but the coding region is different. So is there something wrong? Is there any concerted evolution among those common region, or there's any recombination going on in those common region? And you can use the RAT program I just mentioned, and it will show you this is the different line of different components. And this the y-axis are the similarity of the sequences, and you can see the different component will have very similar cases, and these are the common region, just the common region. And here you can see the similarity are mostly the same, except for a particular case, you can the similarity drop a lot. And so if you see such kind of pattern using red, it means that this part has a very little similarity compared to the other part of the sequences. This will likely mean that this piece of sequences are derived from somewhere else through recombination. In this case, it's likely. And so you can use this kind, there are several different kind of similar programs can do the similar things. So you can see the similarity curve, and you can see the different pattern. Actually, sometimes you can just see the sequence and you will know it. But anyway, this is just another way to visualize that. And if you see such kind of pattern happen, then one way to confirm it is to dissect the sequences. For example, if there's a sequence and you found a similarity for this part is similar to somewhere else, and then the other part is similar to another sequences, you will probably find a similarity figure like this one. Then the best way is to separate the sequence from here to here is number 1, and from here to here is number 2, and then use the number 1 and number 2 separated to reconstruct the phylogeny. You're likely to see this part is similar to one thing, the other part is similar to another thing. And well, theoretically, ideally, it should be doing that, and you will see, okay, this part is derived from somewhere, and the other part is derived from somewhere else. And the case from Sanguinaria I just mentioned is just the case like here. But if it's very easy to visualize on the sequences, it's very easy. But if the sequences itself is very variable, or it's a virus, so its substitution is a lot, then it's very difficult to visualize by your own eyes. So you will need to have some algorithms to really cut a sequence into 2 or even more several different segments, and to reconstruct the phylogeny accordingly. So it's not very easy to really pinpoint the recombination size if the substitution is very high. So just be aware of that.